Biofortification is the development of nutrient-dense crops which can be achieved using one of three different technological approaches. The first approach is through so-called agro-biofortification. Plants absorb nutrients from the soil via their roots or from the air or rain via their leaves. Agronomic technologies can be used to improve this process. This may be achieved by the addition of nutrients from fertilizer application or by foliar spraying. Techniques used to improve soil quality can also enhance nutrient uptake, such as tillage techniques, weed management, water supply practices, pH control, and the use of ash or animal dung to enrich the soil. An example of this approach is the biofortification of rice through foliar spraying using a zinc solution. The second approach to achieving biofortification is through breeding and selection. In this approach, the gene pool of crops is screened for lines with a naturally higher nutrient content. The genetic pool of crops has been used by farmers for centuries to increase certain traits, mostly related to increasing the production and resistance to plant diseases, or to improve the appearance of the final product. Breeding and selection for higher nutrient content is nothing more than adding another desirable trait to this list. An example of a biofortified crop developed through breeding and selection is the orange sweet potato. A third approach to biofortification is through genetic modification. Biotechnology can be used to enhance the nutrient density of crops. To do this, desirable genes from one plant or species, often from bacteria, are introduced into another plant. This technique can considerably enhance the nutrient content of plants. Golden rice is one example of a plant which has been genetically modified to achieve biofortification. A rice variety that has a higher level of provitamin A.